Hello, I'm Shunichi Kashida and co-founder, CEO of xFOREST Therapeutics. On behalf of our company, I introduce our business and a recent update to you, entitled Exploring RNA Targeting Small Molecule for Sustainable Medicine Against the Undruggable Disease. Before talking about our platform, I want to explain what are undruggables. That's more than 5,000 known disease-related genes. However, only a small portion has been drugged. 50% of these genes are considered to be undruggable on the protein level with conventional methods. But we can challenge by the targeting messenger RNA level with small molecule. If we cannot target these disease-related protein, let's target the upstream blueprint of the protein. That's the RNA. As benchmark icon of this drug discovery field, xFOREST from Roche, Genentech, and PTC collaboration showed it is possible. Treats SMA disease and is a 1.6 billion sales blockbuster. It was found in phenotypic screening, but it is difficult to reproduce this success, mainly because of low hit rates and selectivity and optimization difficulty. So, xFOREST provides a unique platform that addresses these key challenges in this RNA targeting drug discovery field. The challenge from the target RNA druggability, throughput of target RNA, and the most important point is the selectivity with high affinity. It's related to off-target side effect. And the binding site in identification, and there is not so much successful hit RNA binders, so there's no knowledge about the target RNA space and the chemical space. With our technology, all the current challenges in RNA targeted small molecule drug discovery can be overcome. How can we do this? The key is our unique and large-scale biochemical HTS to discover quality RNA binding small molecules. Our Matrix Forest platform is an all combination profiling of RNA structure library versus compound library. We start with the mixture of thousands of RNA motifs and dispense them into well plates. To each well, we add different compounds of a compound library. They are freely in solution in a chemical reaction, and it's for the on-plate biochemical assay. We use next generation sequencing as a readout to generate big data sets. This data contains the binding information on all combinations of RNA motifs and the small molecules. Besides the binding information on the order of a billion combinations, additional data is obtained. Because we use so many RNA motifs, the selectivity of binders can be assessed. And the single nucleotide resolution gives us information where in RNA structure are important in the interaction with binding small molecule. This platform and our experience allows us to find a selective binder with submicromolar activity. Additionally, our precision and dense big data is useful for data analysis, including AI. Conventional methods work on a much smaller scale and cannot provide such kind of results. I want to give an example of an advanced collaboration project. In this project, we designed and prepared an RNA library, and we conducted a screening using our Matrix Forest technology. I found more than 20 selective hit compounds and explore the chemical space. In the hit validation stage, we got the hit compounds profile with some micromolar affinity and cellular function, and hit derivatives activities as a SAR information. SAR means the structure-activity relationship. And this is only one case of collaborations. Since our foundation in 2020, we started several collaborations with the Japanese big pharma companies, Takeda, Daiichi Sankyo, Kyowa Kirin, and Otsuka. All of them include licensing agreements. With Takeda, we reached the first milestone last year, and last month, we received another milestone payment from Kyowa Kirin Pharma. And Ulrike Gao, our EU business head, representatively joined the last HVC program. We learned about what pharmaceutical companies additionally need. And from the learning, it is important to anticipate the pharmaceutical demand and complete the process of a lead generation step. So that step is the before animal test. And it means that the addition to hit discovery platform, more downstream lead compound generation platform are demanded. At xFOREST, the hit to lead stage analysis, integrating the matrix forest with RNA specialized analytical methods, and supporting the discovery of high quality lead compounds. And we need to prove the concept of the hit to lead process platform. To do that, we have to synthesize the thousands of hit compound derivatives and acquire the integrated big data by ourselves. Then we got the funding for our hit to lead POC project. In addition to 6 million Series A funding, we awarded a governmental matching fund of 7 million for our POC projects of RNA targeted specialized platform for hit to lead generation process. Now our internal hit to lead compound generation program is ongoing. So we launched the 4 target programs now, and within 2.5 years, we will launch another 10 targets. And 40 to 200 compounds synthesized and doing these SAR for the pharmacokinetics, and also the high throughput ADMET for the pharmacodynamics. These are the analysis every month. And we tried these cycle of the compound synthesis and analysis and the designing. And first, the lead indication and the lead optimization launch, it's the launch planned in the end of this year at earliest. It's a little bit ambitious, but we tried work hard for that. So, and this project will provide us not only this lead compound and this the hit to lead platform, but it's a really good feedback to our original HTS platform for updates. Yeah, and in the horizon from our continuous effort for updates of our own unique technology and these collaborations, we will lead this RNA targeting small molecule drug discovery for sustainable medicines and against the undruggable diseases. Thank you for attention.
Okay, Kashida-san, thank you very much for the great presentation. I have a quick question because I was trained as a protein chemist, and targeting RNA, probably the hit to lead, which you mentioned at the end of the presentation, should be different, especially about, of course, the efficacy should be different, but at the same time, like ADMET talks or like what kind of toxicity we have to search on that. So do you have, can you elaborate a little bit more as far as you can disclose to us?
Okay, yeah. So maybe how to say, comparing to the, sometimes it's the overlap. The important point is the which type of the chemical space is the more, how to say, the RNA targeting. But from the learning, from this receive from what's Roche and PTC and Genentech developed. So a lot of the followers they learn from this chemical space. So there's some very specific some characteristics. So with this narrow space or some other just the RNA binding, instead of usually losing the selectivity of the binding. The important difference between the RNA and protein is that the kinds of the players of this polymer is that only 4 or 20. So if you not consider but this more 3D structure or some other characteristic of the target RNA, you can easily lose such kind of more the selectivity. So that's why the usually RNA binder looks toxic and quite kinds of positively charged. So that part cannot be easily evacuated from this type of follower. But what's our important progress is that we screen the typical protein targeted chemical library we use for the screening system. And then we try to find such a pre-existing QED, or so what's the good chemical property, is that mutually simultaneously meeting this the requirement for the RNA binding. And it's quite rare to find it. But thanks to these 1 billions of combinations of our screening, we can find these rare hit. And from this rare hit, we expand the chemical space. So that's the kinds of another strategy, so that we can find really good, the more predictable, kinds of chemical candidate for these toxicity, and also some finding the good efficacy as the RNA binder and the RNA targeting drug.
I see. Yeah, I had a lot of questions, but limited time, so I'll forward the question to you.
Yeah, thank you very much, Kashida-san. So good learning for me. So in addition to your collaboration model with a big farm, you also introduced that you have your own programs in the pipeline. So are you going to bring those programs to clinical on yourself, or what kind of a business model in your mind?
So in that point, there's our the expertise, the initial HTS level, we can put a lot of different RNA structure from different genes. So in that case, we can leverage this number of such kind of targets. So at the starting level. So that's why the pursuing the single target is sometimes it's a little bit risky. So that's why we try to still this the number of the hit to lead campaign is kinds of a lot for the small startup. Yeah, but we try to just enlarge this pyramid to go further. So once we get the really good hit to lead compounds, we try to outright things and getting the budget, and then we try another level. It's maybe preclinical. So and then once we got for example, doing the IPO or something, become a bigger company, and then we can try the clinical phase. We think. I don't know whether it's a good strategy or not, but at least it's in this field a lot of some competitors in the US try to do this clinical phase, but sometimes it's very difficult to pass to the further. So we have to make a really good basement over the more scientific this RNA targeting small molecule drug discovery field, and then we can build the confirmed such kind of stretch the construct of our business.
Yeah, I really appreciate your step-by-step approach. For my understanding, maybe near term, you collaborate with a big farm if they can name a target, right, for you to work together. And in the middle term, you have your own target identified and develop the program, thinking about the out-licensing model. But in long term, you're going to bring all those program on your own.
Yeah, you're
Yeah,
right.
thank
Yes.
you.
Thank you so much.
Thank you.
Thank you for summarize.
Do we still have a time?
One quick question, please.
Yeah, well, so I have two technical questions. So, so in the HIT campaign, so presumably, so you can identify several binding sites in one mRNA. So my question here is how to select the binding site in one mRNA.
Yeah. And,
Any strategy? If I'm a client, right? Mm-hmm. Then if you identify the many binding sites.
Okay. So sorry, but I skipped our explanation. But at first, we try to kinds of the size the RNA structure motif from the whole length of the messenger RNA or pre-messenger RNA. And then usually we correct these the piece fragment of this RNA from the 50 to 500 numbers. And then once we got the binding hit, the partner target RNA region is already determined. So that's why we automatically find the binding site as a single one. Mm-hmm. If it's not binding to the others in these are the RNA motifs. So and then the convertly, whether this in a total length of a messenger RNA situation, this compound still can bind to this site or not, we have to check in a cell. Mm-hmm. But we already have such kind of a technology.
Okay.
Yeah.
Thank you. And then next question is that so if we successfully identify one site to get the modulator of the mRNA. Mm-hmm. So you said several hit compound in the hit campaign can be identified, right? Within that hit compound, are you able to find compounds in a different structure class or just only one structure class and its deliverables, analogs?
So you mean targeting the single site?
Yeah, single site.
Okay. Yeah. So actually, it's the case by case, but we already found different chemotype. Mm-hmm. That means a structure class already. Yeah. So that's and also some published paper for these analysis. They also found several these compound structure. So that's why it's quite reasonable, I think.
Mm-hmm. Thank you very much. So one last question. So in the protein, so we usually use a crystallography, as Koyanagi-san said. So in this case, so are you able to kind of establish a similar technology to kind of determine the binding mode to each nucleic acid?
I think so. Thanks to our matrix forest system, we can very early stage already determine which is our most reactive site, nucleotide. And of course, once it's the binding affinities become stronger, we can make the crystallography as well as the protein, and checking whether the binding nucleotides is the same or not, that we can check. And even we can try with the NMR as well, I think.
Great. Thank you very much.
Okay, then, Kashida-san, thank you for your presentation, and please give it a big applause. Thank
you. HVC 京都2025スタートアップ起業を志す研究者募集中。 HVC 京都はヘルスケア領域のスタートアップとオープンイノベーションに積極的なパートナー企業やベンチャーキャピタルが国内外から集まるイノベーションプラットフォームです。一流のアドバイザーから海外市場を視野に入れた事業戦略、事業プレゼンのアドバイスなどを受けることができ、事業提携先や共同研究先、出資先を検討しているパートナーとの個別商談を実施しており、多くのマッチング実績があります。詳しくは動画の概要欄のリンクからエントリーシートをご参照ください。エントリーは無料です。創薬、バイオ、医療機器、デジタルヘルスなど ヘルスケア領域のスタートアップの皆様、研究成果を実用化したい起業志向の高い研究者の皆様のご応募をお待ちしております。